consent to being recorded. All right. Hello, everybody, and welcome to our Morsels series, Numbers and Nature Talk. We are excited today to be joined by Dr. Kristen Rappazzo from the EPA. She is a talented epidemiological researcher focusing mostly on methods, and today she's going to talk to us about some of her research investigating potential for bias in health studies. Take it away, Kristen.
All right, thank you very much for having me. I'm going to share my screen, hopefully. There we go. All right. All right, can you see my slides? Awesome.
Perfect.
So I'm going to talk about a project on correlated confounding and the adjustment approaches for confounding using mixtures, methods, contexts. And just to start off with a disclaimer, these are, actually, I'm going to go back and say, so I am talking about it. Hannah Jardell is my student who is leading the project, and she has done all of the work and all of the heavy lifting for this project. And I really love having students. It's wonderful. And then Allison Krajewski, who is a colleague at the EPA, and Alex Kyle, who is at the National Cancer Institute, are co-authors on this project. And then to start off with the disclaimer that these are our views and mine in particular, and not those of the US EPA. So we have these projects because traditionally environmental epidemiology looks at single exposures, and a lot of this makes sense because the way that regulations are written, and if you want to impact public health, you target your research towards what the politicians are actually going to take action on, or what cities, local governments, etc. But there is increasing acknowledgement about what the EPA is now calling cumulative impacts, and what researchers will also call mixtures methods, and that basically we are not exposed to a single thing at one point in time, and that's it. It's not in isolation. It's usually a whole bunch of stuff, often all together. These things often share sources or are embedded in social structures that mean that you are getting worse exposures clustered together, and better exposures also clustered together. And so this is the correlated with potential confounders, such as a lot of socioeconomic status. This is the environmental exposures are not randomly distributed across the population, or we wouldn't have environmental justice issues, which we have plenty of in the United States for sure. So these socioeconomic factors in particular, why people live where they live, and why contaminants are where they are, end up where they are all interrelated. And so we can think about these exposures as themselves a single actor, but also they're acting as a group, and these members of groups might potentiate each other or alleviate each other, depending on what they are. So examples for that, we have air pollution and green space. Like we think green space will control or reduce air pollution. So if you have both of them together, maybe you're not as impacted as someone who's living in an entirely urban gray space area. And there's increasing interest in these epidemiology mixtures methods. I know this is a more mathy group that I am presenting to, so you're probably going to be looking at these methods and be like, oh, so cute. But so some of these are really complicated, and because we are often using observational data or just pre-existing registry data out in the world, we are working with the limitations of available data. And so we understand these methods have been developed, and they're being increasingly used in the epidemiology research arena. And as someone who is in the federal government, and one of my various jobs is doing assessment work, and Allison Krajewski, her main job is doing assessment work for the EPA. And that assessment, those scientific assessments go to inform on policy actions, and hopefully have the better informed policy and scientifically sound policy. That's the goal of the EPA in general. And we understand how these methods, because they were developed by methodologists and statisticians, how they behave in simple, controlled context. But there's also things we don't understand, and these more complicated structures sort of getting into that area of, like, what if everything is correlated super highly? What if it's also correlated with the things you would normally adjust for as confounding? What if one of the things is good and the others are bad? So these are sort of things we want to explore and know about, especially as people who will potentially be doing assessment work on these studies, and understanding the choices that the scientists made and how those impact the results. So there's a couple of examples. So the highly correlated confounders, which is mostly what we're focused on. And there's also these people add what are predictors of the outcome, and they're called precision variables. Ideally, they're supposed to improve standard error, reduce standard error. I am not fully convinced that is the case when people use them. Sometimes it's just, this is a risk factor for the outcome, and I don't want to fully think about the confounding structure, so I just threw it in the model, and I think that's the actual reason they end up in models. So this study, we want to investigate and quantify the potential for bias from different confounding choices, or different confounding adjustment choices, when using two of the more common mixtures methods. So in this case, it's weighted quantile sums, or WQS, and quantile g-computation. And because my history is in reproductive epidemiology for the most part, and we have data on that, real world data on that, and we know more about what is happening or what should be happening, we use preterm birth as our outcome of interest, along with hazardous air pollutants, which are known or suspected reproductive toxicants. And that's our environmental exposure. So we started the project by conceptualizing what should our data look like, and using a directed acyclic graph, or DAG, which a lot of the epidemiologists trained in the more methods-focused schools love DAGs. They're great, so fun. And we based it on empirical relationships that we know about and data that exists out in the world to simulate the analysis data sets. So here's our DAG. You can see ideally, the air pollutants actually should be broken out into the 8 different hazardous air pollutants we used as the mixture, but for simplicity's sake, we had it as a single node. And you can see we have experienced racism is sort of a hidden variable, a hidden confounder, where it's strongly influencing education and income, but it's not itself directly connected. And that's just to better conceptualize why epidemiologists frequently include race in the model. And it's like, we really do want to get at this experienced racism and not the actual color of your skin or how people perceive you, other than how they react to you, and stress that comes along with that. We also have some, in this case, unnamed confounders. We did name them later, and preterm birth is our outcome, and we have some noise variables and a predictor that's only associated with the outcome. We also have an instrumental variable in the DAG. We didn't end up pursuing that because in mixtures analysis, instrumental analysis doesn't really make sense. So we decided to table that one. And then and when I say we, I mean Hannah, for the most part, when I'm talking about the actual doing of things. So we simulated pollutants based on the 2011 National Air Toxics Assessment, and the list of pollutants is there. We had racism as the hidden confounder, which strongly influenced socioeconomic status confounders, and then these other confounders, which are sort of known to be potential confounders in general studies of reproductive health outcomes. And yeah, and we conceptualized the predictor as fetal plasma testosterone. And that was because people adjust for sex a lot in studies of environmental epidemiology, environmental exposures during pregnancy and birth outcomes. And it's one of the things that irks me, and everyone who knows me knows this, so that's why it's in there. And again, because I know it's a more mathy audience, I put in the equation. I'm not going to read it, but it's there for everyone to see. There was math involved in the study, in the analysis. And so simulated 500 analysis data sets, each with 5,000 observations. We also subsequently have done a couple of sensitivity analyses with smaller populations to sort of reflect what people might actually be able to study in the context when they have measurements for these types of pollutants. And our truth was not predetermined, but instead what we did was run the same simulation and data set generating process for a data set of 10 million observations. And we ran those truths separately for both WQS and QGComp. And I will tell you, doing these mixtures methods on 10 million observations will kill your computer, and you need a lot of processing power to do that. Sometimes it even kills clusters, which was a fun thing to find out. And so our analysis is honestly pretty simple. We ran either WQS or QGComp, and then went through a series of different modeling choices. So just the exposures, the sort of ideal minimally sufficient model, which is what the sort of the gold standard of confounding is controlled and you don't have any extraneous variables in the model. And then models where you're like, I just put all the confounders that I think are in there. And then some extra stuff. And then everything I've measured goes in the model, which is definitely less than ideal. So if you ever do epidemiology, try not to do that one. And we looked at the mean absolute error and bias estimates. And again, it's a pretty simple, straightforward. The most of the work was done setting up the data set, and the analysis part is actually very straightforward, which that part is nice. Setting up the data set took a while. And what we have basically is that weighted quantile sum tended to have less spread. It was sort of more peaked in its distribution, and QGComp had more variance. But QGComp was also less biased, and sometimes a lot less biased in the case. Like QGComp was pretty like on the target of the truth value from the larger simulation, whereas WQS wasn't as spread out in its estimation, but it was sort of systematically shifted from the target estimate. These are both things that are interesting to know. And again, this is just showing the variance distribution, and then have an actual picture, which I know Nina would rather have the numbers, but since when Hannah was running this and she came up with this, she's like, I don't understand why it's so weird, why weighted quantile sums has like this, it seems like it's more on target, but also has a lot less variance, but it's also biased. And I was like, well, it sounds like the distributions look like they do here in this picture, where QGComp is more varied, it's more spread out, but it is right at the mean for the mean of the QGComp was right at the truth estimate. And WQS is more peaked, which you can see as in most of the models, the WQS is much higher than the distribution at the mean is much higher than for weighted g-comp. But it is also sort of always just a little off from the truth, the target estimate. And so was like, so that's what we've done so far. And I think it is important that we saw differences depending on what model, like what mixtures approach you used, and also how the confounding choices that were made. And it just highlights that the choices in modeling matter. And I'm sure everyone who wants to listen to this is probably already aware of that. But it helps us, especially in the government, to reinforce that and to have evidence that's like, when we do this, different things happen. So we have to think about that. And when we're at like in potential future assessments of this type of research, it helps us know to like, when I say, this study used this particular method and this is their modeling choices for confounding, we can say, well, this might be more biased than this other estimate, which made different choices. And it's so improving our own ability to stand these complex models. I will also say as a side note, one of the things I feel like we definitely proved was that weighted WQS took a lot more time for the computer to run, even when you have very, very impressive computers and processing power and clusters. It always took a few days to run, and the QGComp was a lot quicker. So for processing, QGComp is definitely an easier choice. And that's all I got.
Woohoo!
I think I was right about 15 minutes too, so yeah.
Yeah, no, that was really impressive. For many reasons, not just because you hit the time mark, but also because really cool science, really cool modeling. So I have to confess that I want to ask Phil